I'm Cath Rees, so I'm a Professor of Microbiology at the University of Nottingham, and as another part of my role is that I set up a small biotech company which was founded on the research that we carried out at Nottingham. Everyone's sort of familiar after COVID with the idea that we have viruses that would infect cells. Well, what we work with are viruses that infect bacterial cells. And what we have used is a part of their biology to help us break those cells open. Now, these cells are very robust. They are the sorts of cells that live in the world and are really difficult for us to break open any other way. But because we've used the virus to do it, they're really efficient at it, and which means we can get the DNA out, and then the whole world of molecular detection is available for us. And what we're using that for is for diagnosing disease with these difficult bacteria. We're looking at this phage, and as I say, the test is called ActiPhage. So this is what we've developed as a company. And the long-term aim is to help TB diagnosis. Now, it doesn't really matter if we're talking about humans, because the way that the test is configured, we could either look for disease in humans or we could look for disease in animals. So things like bovine TB, big issue in this country for certain. And the impact is that we are bringing a new way of looking for the disease. Traditional microbiology has always struggled with these diseases that cause TB, the bacteria that cause TB. And using these tests, we're now sort of being able to get really quick answers from sort of typical clinical samples and get a diagnostic, rather than waiting the long periods of time that you normally have to do to get these, to detect these diseases. Now, what that means is that hopefully we can improve diagnostics, so we can treat faster, so we can stop spread of disease. So it's kind of one new tool which is going to help with getting all of this difficult disease under control. Phage research for a long time was a very theoretical. It was the way in that we got into molecular biology and bacteria. So back in the 1960s, that's what people started on, because they were a nice, simple system. There was little viruses, big bacterial cells, and you could use those to sort of start understanding bacterial cells. And then they kind of got forgotten as its sort of primary thing that was a virus that infects cells. Of course, science has gone on and time's moved on, and antibiotics were developed, and then suddenly antibiotic resistance came along. And everyone's gone, oh, hang on a minute, there were those things back in the 60s that people were looking at. And it's just been a sort of field remembered. And because of all where the knowledge has developed over time, we can take it so much further, so much more quickly now. Because back in the day, it was sort of all discovery, but now we're going, okay, well, we've got all this other science we can use with the phage, and we can really start to use sort of phage in a more directed way, in a more rational way. I mean, when phage first came out, there was one of the guys who sold it said that it could cure broken legs, and you're going, clever? Don't think so. Because they didn't really know what they were. But now, in the modern era, we can take all that knowledge, years and years of science, put it all together, and really move the phage field forward. I mean, the majority is trying to cope with this antibiotic resistance problem we've got, using these phage to kill off bacteria instead of antibiotics. We've taken a different approach. We're going through diagnostics, but it's the same basic idea of lots of knowledge applied to a new discipline. This has been 30 years of research, and it's foundational knowledge. It's all the knowledge that we have looked at this system as an academic question for 30 years, and you've suddenly got to the point where you could use that in a very applied and very real world area to have some impact. And that is, personally as a scientist, to see your knowledge sort of helping mankind or even helping animal kind is great. That is the ultimate validation for us, because we're not driven because we want to get the answer, we're driven because we want to ask questions. And when those questions then lead to knowledge that then leads to something, it's just so satisfying. I say, it's been a long time in the coming, 30 years, but now you see all of that work and where it's, the fruition of all of that, it's really great. There isn't a particular highlight of my career, there is a particular highlight moment of the whole science. The very first time that we got real clinical samples from cattle, and our theoretical test that we developed in the lab worked, and the predictions we'd made came true, and we could do what we said we were going to do, It was just so exciting. It was a moment where I hugged the PhD student. So, you know, just because it is that moment where you think, yeah, we were right. And I don't think you get many of those moments. So when I found out I'd won this prize, I was really surprised. I say, you spend your life going on doing your own path, and you don't really think about how other people are looking at what you're doing. And so, you know, to get a letter to say that actually the society has recognised what we've done, you know, it's really great. Yeah, really surprising, really refreshing, and just delighted, really chuffed. I was genuinely touched that the society, you know, obviously I got nominated, but the fact that the society thought that I was deserving of this, it was really appreciated. But I say it's not just my research. And there have been two people in particular who've been in my lab, which is Dr. Ben Swift, who's now a PI at the Royal Vet College, and Berwyn Clark, who came from industry and inspired us to set up a company who, without them, I wouldn't be sitting here. So thanks for them as well.
It's my great pleasure to welcome this afternoon the winner of the 2024 Translational Microbiology Prize, Professor Catherine Rees, from the University of Nottingham. The Translational Microbiology Prize is awarded to an individual who has demonstrated an outstanding contribution to translational microbiology. I think we can all agree that Catherine meets the criteria. Her research has resulted in the development of a rapid phage-based test, Actiphage, for the detection of mycobacterial pathogens, including Mycobacterium tuberculosis in humans, and Mycobacterium paratuberculosis and Mycobacterium bovis in animals. This work led to the establishment of PBD Biotech Limited, where Catherine was the Chief Scientific Officer until 2022. In 2019, Actiphage was awarded the Royal Dairy Innovation Award for R&D, and in 2021 it received the British Veterinary Association Innovation Award. Currently, the company is developing Actiphage as a rapid human diagnostic test with promising results gained in early clinical trials. Since stepping down from her role in the company, Catherine has joined the Government Advisory Committee on the Microbial Safety of Food and the Advisory Board for the Innovate UK Phage Innovation Network. It is my great pleasure to welcome Catherine to talk to us about the challenge of developing phage-based assay detection methods for mycobacteria, lessons learned on a journey from cattle to humans. Catherine.
Oh, good evening, everybody. So I survived stage one, got up the steps without falling, which is good, because that would be on YouTube. So I'm going to talk you through the sort of my life experience, as it were, as we went from fundamental research into the development of a product. And it's been really gratifying that the Society has chosen to recognise what we've done. I was interviewed earlier and I was saying it's weird because you're kind of there in the middle of your own thing, just getting on with your life, and you don't realise that other people actually take a notice of what you're doing. So it's a great honour to be here, and I really appreciate the award of the prize. I'm going to mention a few key characters as I go through, because this has not been a solo effort. So we're going to talk about the challenge of developing the phage-based assay, and lessons from the journey to cattle to humans, but actually the title should be From Humans to Cattle to Humans, as you'll see. So let's just set the scene, because I know you're not all mycobacterial people in the room, so I'm just going to talk to you a little bit about why mycobacteria are important to us. They're responsible for major intractable human disease, TB, okay? Now, the graphic at the bottom there is a nice little infographic that I took from the WHO NTB strategy, which was in 2015, so you can see all the dates of 2014. But in reality, in the current version of that strategy, which is the NTB by 2035, because we didn't quite get the targets in 2020 or 2025, those numbers have just gone up. We haven't actually got to the point where we are controlling this disease. Now, one of the biggest problems is that you get spread in the community, and diagnostics is part of the issue, because the primary diagnostics is someone presents in the clinic with the symptoms of TB, so with fever and coughing, and you have a chest x-ray, or you have to do a test to look for an immune response to the bacteria, or you try and look for the presence of the bacteria in the sputum that they are coughing up. Okay, so it's not a nice, easy diagnostic. It is all sort of secondary, because we can't do the microbiology that we'd like to do, the sort of quick test, the culture, the identification of the organism. So let's just introduce you to our friends, the Mycobacteria then, rod-shaped bacteria, genetically related to the gram-positive bacteria. I usually say this for my undergraduates just so they know where they're fitting, because they are so weird you'd never believe that. So we're thinking of sort of Listeria, Staphylococcus, lactic acid bacteria, which we can handle in the lab, we can grow them in 24 hours, it's all fine. But when we get to the mycobacterial pathogens, they're characterized by these really slow growth rates. And I've often have to point it out that that is not a typo. It is 8 to 18 weeks for cultures. So some of my PhD students would put their plates in the incubator and come back 3 months later to see whether an experiment would work. So inevitably, research is a little slow. It's not like working with things like E. coli or Listeria. Okay. Now, because of that slow growth, it means that the culture-based method for diagnostics are just impractical. You can't leave someone that long before you go back to them. And one of the characteristics of things, I say, these are in that sort of group of gram-positives that makes them so different, is their cell wall. So they have this very distinct cell wall structure, which is waxy. It's got lots of mycolic acid in it. It almost forms an outer wax layer on the surface of the cell. Now, this is the sort of typical textbook diagram of it. So you can see here's the peptidoglycan layer, and then all of this, okay, is this wax up here. Incredibly difficult to penetrate. But that means if we want to use our other favourite tool in terms of diagnostics, which is PCR, you've got to break the cells open. And they are incredibly resistant to the sort of chemicals that we use for standard lysis. So unless you've got really high cell numbers, if you've got an efficiency of lysis that's only 1% of your population, unless you've got lots of cells there, you don't get enough DNA to do PCR even. So they do create some real challenges for us in terms of diagnostics. And they're also difficult as far as their immune response is concerned. So mycobacterial diseases are characteristic by the fact they develop slowly, and the lesions in the tissues only really become evident at the late stages. So on this oops, I'm pointing at the screen, that doesn't help. Pointing at this diagram here, sort of clinical symptoms appear here. This is where we're starting to see sort of lesions in tissues that you can identify that are characteristic of disease. So the other problem we have is these are intracellular bacteria most of the time inside when they're in their hosts. They're living inside our cells, so they're not easily targeted by antibiotics. And they've also, like we heard this morning, developed systems where they suppress the immune response of the host, so that they hide. They're sort of sitting there quietly saying, you can't see me, I'm not here, don't worry about this. And so we get this delayed antibody response. So basically, if this is at point of infection, this is time, we've got antibody responses, but until you're really getting to the point where you're seeing significant clinical symptoms, even antibody titers are very low. So it really makes early diagnostics difficult. So, as I said, often only seen in the late stages of infection. What we know is that over time, slowly, the bacterial load is going up, okay? And eventually that results in lesions that are visible in the tissue. But we have this sort of long latent period where the host is infected, there may be some symptoms, but there's nothing really we can do diagnostically, and you've got to wait and see what's happening. So there's the mycobacteria, there's the problem. Intractable disease, diagnostics limited or difficult because of the nature of the organism. So my original training was in bacterial genetics, and when I was a youth, phage were the best tool we had as far as bacterial genetics were concerned. So we were using phage for lots of different purposes. And so you were introduced to phage already in these talks, but I will just remind you that these are viruses that specifically infect bacteria. So when we're looking at an agar plate here, we can see the zones of clearing where all the bacteria are uniformly growing across the surface of the plate, but where the virus is, we can see these zones of clearing where the bacteria phage have killed off the bacteria. Some electron micrographs there of some phage. This one is a beautiful image, came from a group in Finland. This is helium ion microscopy, and you can see there's the bacterial cell surface with these viruses attaching to the surface using these tail-like structures that give them specificity. Those viral particles have receptors in the tails that means they can recognize their correct host. But once they have infected, like all viruses, they take over the host cell, they replicate, make lots of copies of themselves. But now they need to get out. Okay? Now we're not in a eukaryotic cell that has a relatively sort of permeable cell wall or fluid cell wall. We're in something with a nice big rigid structure. So these viruses have evolved enzymes that specifically target those cell wall structures from the inside out. They're going to target the peptidoglycan and to break open the cell wall and then result in cell lysis. And you can see this massive explosion here with the small viral particles coming out. So, in sort of terms of viruses, they're very typical of viruses. But what we are going to take advantage of the fact are those two things, the specificity, that they can recognize their correct host, and the fact they're great at breaking open cells. So I'm going to go back to the beginning of this project, and it is a 30-year project. When I first joined Nottingham, I joined Professor Gordon Stewart's lab. Gordon sadly died too early in 1999, and he's still missed because he was a great mentor, a great friend, and a really inspirational scientist because he really thought out of the box. But he had a background, he'd come from industry, and he was interested in translational science. And he was involved in the development, or he led the first development of the first phage test. It was the 1980s, it's a little bit 1980s in colours of the box, okay? But this was a commercial test, it went through all the sort of validation you need to do. But it was a microbiological test, and what it was looking for was to detect TB in human sputum. Now, I'm going to explain the principles of this assay, and quite often when I give talks, I gloss over this, but I thought, okay, I've got a captive audience, I'll go for it. This assay now was badly named, because the name that they gave it was the phage amplification assay. Now, when I say they gave it, I was in the lab but not working on this project at the time, so I think I might have objected. But anyway, phage amplification assay was the name. So let's start at the beginning. You take a sample of sputum from a patient, and there are lots of methods that have been developed to separate that horrible mucus, to separate the bacteria from it. So decontamination processes were already well known, okay, for culture, for microscopy. So we used the standard methods. So we get to the point now where we've got a liquid sample, where we've got the bacteria separated from the mucus, and you add a virus, you add a phage. Now, the critical bit of this is that you only leave it for an hour. Now, if you're working with a fast-growing bacterium, an hour is probably sufficient for the phage to infect, replicate, break it open. Because these things are growing slowly in the lab, that's long enough for the phage to get in, but they are not yet going to come out. And that's a really important part of the assay. So we leave it for an hour, and then we add a chemical, which is a viricide, which anything that has not infected a host cell is destroyed. So we chemically inactivate any phage that has not now been protected because it's injected its nucleic acid into its host. It's done the recognition, started the infection process. Now, the other feature of this assay is that it uses a very broad host range phage. Because of the difficulty of growing mycobacteria in the lab, most of the phage we have for these low, slow-growing pathogens were originally isolated on non-pathogenic environmental mycobacteria that grow relatively, not quite as fast as E. coli, but relatively quickly in the lab. So D29, our phage of choice, infects both the pathogens and this fast-growing bug. So you add in your fast-growing bacteria, you pour it into a Petri dish, and you leave it overnight. So anywhere there was an infected host cell, that phage was protected. It breaks open, does the explosion bit, releases new viruses, and a plaque starts to grow on the lawn. So you get to the situation then, as I say, they should really have called this a phage protection assay, not a phage amplification assay. But you have the situation of this equivalence of each of the plaques represented one phage that was protected in the original sample by a mycobacterium. It can only be protected by a mycobacterium because it's specific. So we've got this equivalence between plaque numbers and cell numbers in the original part of the assay. Okay. Now, the other nice thing about D29, it's one of those viruses that is very, very sensitive to the metabolic state of its host. It just does not like infecting slow-growing cells. So we also get a readout of this of actively growing cells, okay? And that was the basis of the original test. You counted the number of plaques. And if you were healthy, you may have maybe up to 10 to 15 plaques on a plate because mycobacteria are everywhere, not pathogens, just environmental organisms. So, yeah, there was some protection going on. But if you're infected, the numbers go up massively, hundreds of plaques on a plate. There you go, that's an infected individual. Okay, so that was the original format of the test. But round about when that kit came out in its nice box, PCR came along. Even if you've got, slowly grown, concentrated samples, you can eventually do PCR from sputum. You've got enough cells in the sputum, you can do PCR. So the diagnostic test in humans went down a completely different route. It went down, let's just take heavy contaminated sputum, PCR it, and we'll see if there's TB there. But the company still owned the technology and said, it's a good test. Can we do anything else with it? So that's when they came back to Nottingham, and at the time I was the PI there leading the phage group, and they said, well, can you do anything with this technology? Is there anything else we can do with it? And the first target they were interested in was Mycobacterium paratuberculosis. Now, unless you work in agriculture, you may not have heard of this organism. It causes Johne's disease in cattle, which is a wasting enteritis. This poor animal here, you can see every bone on its body. Basically, the gut surface becomes so inflamed they can't absorb nutrients, they starve to death. It is primarily an economic disease because it is endemic in herds, and you get loss of animals prematurely, but it's not actually notifiable or anything else as far as they knew, so it was something that people lived with. But round about this time, there'd been a question raised whether this disease looks an awful lot like Crohn's disease in humans. Could paratuberculosis be one of the organisms that causes, it triggers Crohn's disease? Now, this raised a concern for the dairy sector because they had this endemic disease. It was in the food chain, in the milk. And, if that link was proven, then that meant, in terms of due diligence, they had to do what they could to get rid of it. So the question is, could we repurpose our phage test now for paratuberculosis? And really, at the first point, they were interested in milk because that was the route into the humans. So they sponsored students, and in our group we developed a method. Now all this is exactly the same here in the middle, that's all the same. But now we start with a bottle of milk, and because we know these things are intracellular, like the TB, we collected the somatic cells by centrifugation. So you took the somatic cells, broke them open, lysed them using osmotic shock, release the mycobacteria into some media, add the phage, wait an hour, do they get the plaques? Okay, great. But mycobacteria are everywhere. And one of the places they are a lot are in our guts, and they're also in the guts of cattle. So when you get your number of plaques back, you're going, oh, that's a lot of plaques, because you can get fecal contamination of milk. It doesn't mean the cow is infected, it just means it's not very clean milk. Okay. So what are we going to do? So what we did, and this was a moment of, no, this won't work, but let's try it. So we used a punch and we excised the DNA from the plaque. Now, when viruses grow, the cell that was the first protecting cell is right in the center, and the virus diffuses outwards and the plaque grows bigger. But the DNA of the original cell is right in the center, which does make it easy to find. So punched out the DNA, the agar, used standard DNA extraction techniques, did a PCR using signature sequences for paratuberculosis, and it really worked. Now, I think I was probably as much as surprised as my PhD student, because this thing has been in the incubator for 37 degrees overnight. The cells had broken open. They'd released all the cell contents. There was nucleases. There was everything. Surely the chromosomal DNA of that one protecting cell would not be preserved. It is. Remarkable. Okay. So we published this back in 2007. And because we were doing para-TB, but we'd already had our interest in TB, we showed you could actually use it for para-TB and TB. And by PCR, you could tell which one you were detecting. Okay, so the company was happy. We went to the dairy sector. They said, yeah, but milk's too late. Let's get it out of the animals. Let's do a diagnostic test for animals. Can you do this in blood? You go, oh, right, okay, fine. Right, so another PhD sponsored, another project, and we spent a lot of time working out how you could separate out the mycobacteria from a whole blood sample. And it turns out that there is something in hemoglobin that really inhibits phage infection. I'm not sure the basis of that, but hemoglobin in any sense is really bad news. So we went down standard purification techniques using phycol, took the buffy coat layer, so we got all of the white blood cells, and then broke those open, because remember, they're inside the white blood cells, add the phage, get the plaques, do the PCR. So the assay is exactly the same. And again, we could publish this and show that we could detect paratuberculosis in the blood of animals within 48 hours. Now, I'm not going to show you a lot of data tonight, but this is just one of the studies that we did in that paper, one of the sections of this study. And this is just to show you that when we took the animals, we went to a farm, they were using milk ELISA as a screen. And they knew the milk ELISA was not overly reproducible, or it had to sort of, it fluctuated positive, negative, until it eventually went positive all of the time, and they knew those animals were in trouble. So those are the red animals there. An amber one is one that's flip-flopping, and a green one is one that has never had a positive ELISA. We did blood ELISA in parallel, and you can see even there's not great concordance. It's still flip-flopping backwards and forwards. But all of the ones down here, the ambers and the greens are all negative. But when we did the phage PCR, it was quite interesting. Not only could we find paratuberculosis in the blood of all of these animals, we also found it in one of the ambers and one of the greens. So it suggested we'd gone further down. We've got this more sensitive way of detecting an earlier stage of infection before the antibody response had got going. Now, we also tried culture, but when we did this, the number of cells that we got, probably 50 to 60 at most, to do culture from a milk sample, the limit of detection is 100 cells. So we expected them to be all negative, and they were all negative. Okay. So we've got a test that doesn't agree with anything else. Okay. Is it reproducible? And what we could see is if we took blood sample A and blood sample B, you get a nice correlation until you get to really low numbers. But that's not really surprising. I've got 50 mils of 2 mils of blood, and I've taken it randomly for an animal. If I've only got 1 or 2 cells per mil, the chances of them all having equal numbers, just on a stochastic basis, is limited. So it was a bit bouncy down here, but that didn't worry us, because, once you get to high numbers, it looked rather nice. So let me just talk a little bit about now, put our commercial heads on. This is all very well as a scientist saying, well, I see this as a stochastic variability, and isn't that nice? But what happens if you're going to try and sell that test to somebody? Well, if we look at sort of classical test parameters, this is what we expect to see. So here's our test value increasing along the bottom axis, whatever it is. And we should have lots of zeros because these are our negatives. Maybe they have a few higher values, but you get to a point where a negative is a negative. And you go to the positives and you're going, okay, we've got a lot with a low value, some with a very high value, but we get a distribution of results. That's the ideal. You put your cutoff in the middle, which is what they did with the plaque assay, 20. Above that, you're infected. And that should give you a sort of indication of the result. However, never looks like that, does it? It really looks like this, with this big overlap in the middle. And this is when we're talking about false positives and false negatives. So when we're talking about our test, we're trying to optimize sensitivity. So that's the lack of false positives. So that means anything that's in this group should be on the right side of the line and give me a positive value. Anything that is a non-specific result, so you want a lack of false negatives, as in these ones that have come up positive should not, false negatives are down there. That should not give you a result, okay? You try and set your cutoff to maximize your false positive, false negative values to whatever the application of your test is. So you can move that line around, to optimize it. Now, one thing we realized with our test, given the format, that we had to get a virus, it had to find its target cell, it had to replicate, we had to get DNA, we had to extract it, and then we had to do PCR. Actually, false positives were virtually impossible, and we did prove that. You can put in an awful lot of DNA into a sample before randomly the DNA overlaps with the plaque. So it means our test is quite good because although it may occasionally not be sensitive, we may miss some, it's not going to give us false positives. Okay. So, changing tack, changing organism, we're going to Mycobacterium bovis. So this causes tuberculosis in cattle, a wide range of other animals, but also is a problem in humans in areas where they live side by side with animals. And in cattle, okay, the screening is usually by an immune response because everything else doesn't really work. And what they do is to do a tuberculin injection. And in the UK, we use what we call the comparative test. So this is a vet. He's measuring how much swelling you get after some tuberculin has been injected into an animal. And it's tuberculin from TB, or M. bovis, sorry, and also from Mycobacterium avium, which is the same group as paratuberculosis, which is endemic in this country. Okay? And you're asking the question, is the lump for M. bovis bigger, and the range is 2 to 4 millimeters, with a pair of calipers, is it bigger than the avium lump? Okay, so it's not the easiest cutoff to look at. Now, the pictures at the bottom here are just to give you a bit of a quiz, because these are all animals we have used our test on, because we're not looking for immune response, we're looking for the bacteria, so I can do it in any species I can get blood from. And we did it in sort of badgers and deer and a lion down here. And anyone who knows that's a bongo. And we had to look it up when the blood sample arrived in the lab as well. Okay, so why were we focusing now on bovine TB? Because in the UK, it's a real problem. So the UK government spends about £70 million a year on a disease control programme. It's estimated there's probably another 50 million of losses to the agricultural sector just caused by the general disruption of this disease in terms of trading and livelihoods. And it's clear that the current skin test programme is not clearing TB. Now again, this is a historical graph. You can see this is the percentage of herds in the UK. It's split up by Wales, England and Scotland that were infected. And prior to 2001, it was about the 1% mark. And those of you who are old enough to remember what happened in 2001 is we had foot and mouth. And there was an awful lot of cattle killed and a lot of cattle moved around. And those 1% clearly got spread around the country because we went from this situation up to this. Okay, and it stayed persistently up there. Now, Scotland didn't have the same issue. They've stayed pretty much TB-free. But this means with this numbers of around about 6% of herds infected, we're slaughtering 40,000 cattle annually based on that skin test. And it's not going down. So could we help? Could our blood test for para-TB find TB in the blood of cattle? And the answer was yes. So we managed to get access to some blood samples from animals. Now, in the UK, it's not legal to test animals that have TB until they're dead. So we had to follow them to the slaughterhouse and took blood from the carcass post-mortem, And also, as part of the TB control program, the carcasses are inspected and the vets look for lesions. How far is, this is a positive skin test result, how far has that disease gone? So you're looking at visible lesions and non-visible lesion animals. And using our little blood test, in 24 hours, we knew that 85% of those with visual lesions were positive and 57% of those which were earlier stage of disease, no visible lesions, were also positive. So we got evidence then that we could use our test as part of a TB testing program. Now, the other interesting thing was this was really low level. So we're looking at sort of 30 cells per 2 mils, even in those animals with visible lesions. So it's really low level. But that caused a problem, because now this is in disagreement with the dogma for the disease. We are screening these animals on a farm regularly. We're going in every sort of couple of months and picking out those animals that go positive. And therefore, there should be early detection, okay? Now, this is through a lung infection, it shouldn't yet be disseminated. So the fact we were finding it in the blood upset people a bit, because it disagreed with the dogma of the disease. Okay, now let me introduce to J.B.S. Haldane, for those of you who don't know him. Radical thinker, born just before 1900, died in about 1964, but he had some very advanced thoughts. And this is his 4-stage acceptance of a new idea. And it goes along the lines, I have to put my glasses on to read this. This is worthless nonsense. This is true, but quite unimportant. This is interesting, but perverse. I always said so. Okay, fair enough. So at this point, we thought we were down here. This is true, but quite unimportant because it doesn't fit the dogma and no one really knows what to do with the data. Okay. Now, things were going on in the lab at the same time. And you can remember that original schematic with the phycol and the stages and the agar. It was all a bit much. But what happened in the group at that time is that we turned this into a rapid test. So the blood bit stays the same, but now we just put the phage in, wait for 3 hours, let them do that, release the DNA, filter it. If you're intact and the phage has not lysed you, you're up here, free DNA goes down here, clean it up, do a PCR. So we've turned our assay into a phage as a lysis reagent in a standard molecular detection test. Okay. So we showed that identification was possible within 8 hours. We knew that the phage only infected viable cells, so we're not detecting dead cells, which is always a problem with the chemical extraction of DNA. These are viable cells. The limited detection, we've got down to about 10 cells. In this format, it's now high throughput, and therefore, it's now patentable, and we can set up a company. So let me introduce you to the 2 key players in this, and this is Dr. Ben Swift, who's now a PI at the Royal Vet College, and it was mostly his work. And at the time he was doing the other study, he was developing this method, and he showed that when we used this new method, we went from 85 to 100% in the visible lesion animals, and from 57 to 93% in the non-visible lesion animals. So we'd increased the sensitivity of the assay. Negatives were still negative, and so we didn't lose any specificity. And the other person who came on board about that time was Dr. Berwin Clark, who was in the original company, who'd coming to us say, can't you do anything? The company changed its mind, decided they wanted to not do anything else with these assays, but they were happy to sell on the technology or sell it back to us. And so we put the patent in 2014, and the patent wasn't actually granted until 2019. That took a long time. The paper, therefore, was delayed in 2016. We had all this data, but we had to sit on it till the patent came out. But on the basis of that, we set up our little company called PBD Biotech. I know it's awful to say. Phage-based detection, that's what PBD is. Okay. And with 60K of grants, some from the university, some from East Anglia Agritech, we set up our little company. And we had a new website, now nice and modern, with cattle on it. And we had a new kit with a new box. And that, was all sort of moving forward in that way. So, all right. So far, all we've said is, if you've got a skin test, I can also see TB in the blood. It confirms what we already know. And the skin test is cheap and easy. So where's the value? So could we move the diagnosis earlier? And that was the big question. So this is Dick Sibley, who's a very forward-thinking vet. He's upset more government officials than I can ever imagine, because he likes to challenge what's going on. And he said, OK, let's have a go. So he set up a trial. He did everything that he had to do in terms of biosecurity because of the badgers. Also, he started to look at what we call the single intradermal reactors. So let's not worry about the avium lump. Let's just look at that bovis lump and ask the question, is that reaction to that tuberculin from M. bovis because the animal is infected? He also did TB ELISAs, and we did the phage test in parallel. Okay, now the results surprised us. Now, for legal reasons, okay, these animals are not positive for TB. They have failed or passed the TB test, the skin test, because the avium lump was too big. So on this occasion here, when we first went in, there was about 35 of them. And the orange bar shows you how many of those gave you a positive active phage. So these lumps were, in our eyes, genuinely infected. But all we've got is a PCR. And I know, because I'm a scientist, false positives are very unlikely. But, people don't really believe you. So second time we went in, we cultured alongside, and 9 out of 20 of them were culture positive. Now, given that, there's really low levels, remember, we're looking at 30 cells here, it was surprising that even 9 out of 20 were culture positive, but we could definitely find the organism in those samples. So what we saw was an opportunity here now. We said, look, you could go onto these farms. We've had TB. You can find the ones with the single lump. You can do an active phage test. If they're infected, just get rid of them. Get rid of the TB off your farm. Okay? So we could increase the specificity of that one lump test. Okay, but time went on. We were gathering more data. And you can see that the numbers were starting to go down that we were finding, but they were also removing animals from the farm. So we were getting rid of some animals that they suspected were infected. But at that point, they had their first clear TB test. All the animals passed the skin test. And so work has to stop, because legally you cannot test once a farm is clear. Okay. But we didn't know that. And we'd already done the test, and we knew that there was still TB on the farm, so it was a bit of an issue. So, time has moved on, the company's moved on, the TB test got in the papers, and government got involved. We started doing some work with exotic species, that's where the lions and the bongos came in, and because of that, there was more investment in the company. Discussions with the regulators started, and they said, okay, well, we'll let you go back on the farm, because in July 2017, the farm had gone TB positive again. Well, surprise, surprise, okay? But we need you to do a gamma test. We need you to do that gamma interferon test, because that's our gold standard. Now, the farm was braced for themselves. They're going, oh, well, skin test is bad. Gamma's more sensitive. It's going to be worse. So this is what we found in December 2017. We did this. there were a total, you have to add it all up, no, I think it's 38 in that group there, okay, that had a lump. Those are my SIR animals, okay. So the lump was always the highest number. In the phage, 31 of the 38 were also positive for TB. So we're going, okay, they're positive. Gamma, 3. Okay, well, what is going on now? Okay, now, again, just to finish this little bit of story off, they went again clear there in 2018, and DEFRA again said, no, you've got to stop. So this has been a bit of a start-stop project, and it's made it very difficult. But let's go back to what I showed you at the beginning. Now, this is the DEFRA's version of this graph, and we've got this early reaction with the skin test. So this is cell-mediated immunity up here, and we've got an early gamma test, and my screen's just gone off here. Is that because I've run out of time? No, it's back again. Right. Right. Okay. And so if we've got early stage animals, they should all be antibody negative. We were doing antibody tests. They were indeed antibody negative. They went to the slaughterhouse. There was no lesions. Fine. That's fine. We've pushed it back earlier. There are no lesions on these animals. They had this early reaction to the skin test. So surely gamma should be really high, because that's where it should be. So the question we're posing is, actually what we do is to go onto a farm and look for a really strong reaction to an antigen. Are we actually selecting for animals that are really bad at the skin test? Over years and years of selection, we've gone on and said anything that gives you a really strong response goes to slaughter. We'll keep the other ones that are just a bit borderline, and we'll breed from those. So we're worried now, and if you look at the data for the skin test, the sensitivity of it, in the UK, it has plummeted. However, in terms of the eyes of DEFRA, we'd gone a bit back down this way, so it was all great. Now then, okay, we did, though, have long conversations with government, and it is part of doing all of this. You have to work with the regulators, and in that case, now, we managed to get them to agree that in special circumstances, with permission from the Secretary of State, you could do testing other than the skin test, and actiphage was actually mentioned on the government web pages. Right, I am running out of time massively, aren't I? Yes, this is horrible. Right, okay, so I'm going to skip through an awful lot of this very quickly, and I apologise. But let's go back to the human TB problem, and one of the issues that the TB global problem has come up with is the fact that we need better tests. Now, I'm fortunate that where we are, in the middle of the UK here, the next county down, we have the TB Centre at Leicester, and Pranab Haldar there leads a team who had the remit to look at new diagnostic tests. So, working with him, we did kind of the same thing as we did with the cattle. He had people coming into the clinic, and he asked the question, we got the TB positives, TB negatives, could we detect the TB in the blood? And yeah, I'm not going to go through labour all of this, but yes, we could. All the negatives were negative. 11 out of 15 were positive. The ones we missed tend to be the ones with lower markers of disease. So they had not a positive sputum smear. They had lower inflammatory responses. Culture took longer, so there was lower numbers of bacteria there. So it all looked great. It looked like we could do the same thing in humans who'd been diagnosed with TB. Okay. But the thing that upset people was that these were all immunocompetent. And we're not supposed to have that in the blood. Now, looking at the literature, this kind of made sense because the sort of model of TB has moved on from a granuloma where everything is walled off to a dynamic model where we get the macrophages that are infected moving from the center, picking up bacteria, coming out and getting into the peripheral bloodstream. So actually finding TB in the blood of immunocompetent patients kind of fitted with the modern models of what was going on. But what was really exciting was the fact that when you have a TB outbreak, you also test the contacts in the house. Now, they're doing the gamma test, okay? So this IGRA test here is that gamma test. And what we looked at was what happened in that group down there. Now then, so negatives were negatives. Now we had a bigger group now of negatives. We had all the IGRA negative groups, so that was quite good. So our negative control group was better. And 3 of them gave us a positive phage test out of the 18. There were household contacts, no evidence of disease, but they had then a gamma test positive result. Now, they were all healthy at baseline, no x-rays. And of our 3, 2, and only those 2, developed TB when they were monitored after 6 months. And when they sequenced the isolate that came from these, it was the same isolate from the primary patient. So now we seem to have something that can monitor that transition from infection to active disease, or exposure to active disease. So this is basically saying this status now is where we can use active phage. Now this diagram also shows you that this is what we think about in terms of TB. You get exposed and then it may progress, it may go back again, it may go through different stages, but there is this long latent bit in the middle. And what we're hoping is that we can now get our diagnostics into this stage before you got into active TB and maybe help control disease spread in the community because we can get to those early markers. Right, so we tried to publish it, upset people. We got as far as here, but again, because these were immunocompetent patients, they were not happy. Okay. Because the dogma says they shouldn't be there, but it seems dogma has a very short memory. So during lockdown, we got students doing literature reviews, and you go back through the literature, back to the 1980s and beyond, and you can find lots of studies where they were culturing TB from blood. Now, they could culture TB from HIV-positive patients because they don't have a good immune response, they can't control the disease, fine. But 50% of the studies had control groups who were HIV negative, and they were also getting positive cultures. So it's not just us saying it's there, there's culture evidence as well. Now, I really don't have time to do this. So this is when I had to learn about industry and business plans, and Porter's 5 forces of analysis. And basically the company got to the point of saying, look, this is too hard. The existing test is there, there's no market for it, we can't do that. If we look at the TB market, everything is good, let's go in that direction. So they walked away from animal testing just to focus on human TB. Now, that also meant that we needed some more evidence, so the second trial was set up by Pranab in Leicester. And again, we needed something else that says these positives were really positive because people just didn't believe it. So what he did was to use PET-CT. So he said, I'm going to look for sites of inflammation. So now he's just looking at asymptomatic patients. We're not going through TB diagnosed ones at all, putting them through PET-CT, and then locating sites of inflammation and going in for culture. And when he did that, you can see here, all of the ones that came out, we had 7 gave us a phage positive result, and they were also 4 out of 5 of them where he could get to the site using a biopsy, he could culture TB from that site. So active phage in the blood, again, is really telling us that that is a real active disease. So we're starting to stratify our quantiferon results into those that are exposed, those that are progressing. Okay, so again, that's just summarising what we've got to. This is the limitation of the existing tests. This is where we've got tests that work. This is where it's missing. This is where we think active phage, it'll never go down here. We can't find them all, but we can maybe move it a little bit earlier. So, at that point, they'd been focusing on cattle, but they got another investment in the company for 2.4 million, but we realised very quickly that that is going to go through that money very quickly. So getting the test for human TB, the plan is now to do the work we need to do to finish this off and then sell it to a big biotech, because the cost of getting this to the validated end is just too much for a small company. So, just to finish, Friday afternoon experiments are always worth trying. Just because you don't think the DNA should be there, go for it, all right? It's always worth a try. Don't let dogma persuade you that the question's not worth asking. But remember, changing dogma is quite difficult. Translational science is all based on good science. When you move into the business sector, it's sometimes difficult to allow you to answer all the questions you want because you just run out of money. And translational science is ultimately that validation. We have done all of this knowledge and we can see the impact of it. It's great. And it does require specific skills and knowledge. And to do it successfully, you really need to focus on it. And as an academic, that's why I stepped back 2 years ago, because I know I didn't have the skill set or the time for what this needed. So there's always acknowledgements, and I've given you the key people. I'm hoping that everybody who I've talked about is on here. But Ben and Dick and Pranab certainly couldn't have done it without them, and their enthusiasm and their willing to challenge the dogma really made a success of the project. I'm sorry for running over, but thank you very much.